a crime scene. There was a fight that left a victim dead. You find several pieces of evidence that may contain the criminal's DNA. The victim and the criminal struggled, leaving DNA evidence under the victim's fingernails. After the struggle, the criminal ran out, striking his leg on a table and leaving some blood. You also find a hair that did not belong to the victim. The police identify 4 suspects who could have committed the crime. How can DNA technology help identify the criminal? Our genetic material defines who we are, and the information stored in an individual's genetic code can be used to identify that individual. Scattered around the genome are sites of repetitive DNA sequences called short tandem repeats. At this site in the genome, the sequence GATA is repeated multiple times in a row. 99.9% of the human genetic code is identical between individuals, but the number of repeats at tandem repeat sites is highly variable between each individual within the population because copying errors are unusually common at tandem repeat sites. Individual number 1 has 15 GATA repeats, while individual number 2 has 50 GATA repeats. While the number of repeats in the tandem repeat sequence differ between individuals, the DNA that surrounds these repeated sequences is identical. By designing primers that are complementary to these identical sequences, millions of copies of the genetic material between the primers can be made using PCR, even when the starting amount of material is very small. The amplified DNA can then be analyzed with a technique called DNA fingerprinting. To determine if one of the suspects committed the crime, we can compare the number of repeats at 3 different sites using the DNA from evidence collected at the crime scene and DNA from 4 suspects. The FBI uses 13 different repeat sites in their test for criminal identification, but even 3 sites are enough to give strong evidence for a match. First, the DNA samples are amplified with PCR. Gel electrophoresis is performed on the amplified DNA. The smallest pieces run through the gel quickly, while the larger pieces run through the gel more slowly, so the DNA sequences with the smaller number of repeats end up at the bottom of the gel, while the DNA sequences with larger numbers of repeats remain towards the top. When we compare the banding patterns for the lanes containing the DNA evidence against the lanes containing DNA from suspects, we see that the evidence matches suspect number 2. Suspect number 2 was taken into custody as the investigation continued.